Hello all, my name is Haider Ali, and my research this summer focused on the structural and mechanical features underlying migratory plasticity in breast cancer spheroids. The invasion of cancer cells from a primary tumor is a process that depends not only on the cells themselves, but also on the extracellular matrix or ECM. The ECM is a complex 3D network of proteins and glycoproteins that undergoes extensive remodeling during tumor progression. In particular, increased density of collagen at the tumor stroma interface is thought to promote breast cancer invasion by altering tumor microenvironment. Recent work supports the notion that cancer cell invasion in the form of collective multicellular strands or clusters is the most effective route towards metastatic disease. Previous data acquired by our PI and others show that collective invasion is favored by high collagen density, while single-cell escape is observed in low collagen density. This summer, we followed up on this work by forming breast cancer spheroids from the highly invasive MDA-MB231 cell line and embedding them into 3D collagen matrices. We then imaged them and performed a compression test to quantify the structural and mechanical properties of extracellular collagen that might underlie the transition seen between the migratory phenotypes. As you can see in the schematic, MDA-MB231 spheroids were plated in a low adhesion 96-well plate and allowed to form for 48 hours. They were then embedded in collagen gels to monitor invasion. On the left are two previously collected spheroid invasion time-lapse movies in 2 and 4 mg per ml collagen gels. In the figure on the right, you can see equivalent radius of the spheroid in the 2 mg per ml shoots up due to this fast single-cell invasion, while that of the spheroid in the 4 mg per ml gel increases steadily but at a slower rate. This stark difference in equivalent spheroid radius is reflected by the differences in migratory modes. The panel of multiphoton images shows that single-cell invasion is favored in low-density collagen gels, so both 1 and 2 mg per ml, while protruding arms emanating from the spheroid are the primary mode of invasion in the higher density collagen gels, so 3 and 4 mg per ml. Because we are in the process of setting up a new lab, we decided to start by ensuring that the spheroids and results we obtained were consistent with the PI's published research. To do this, spheroids of 1,000, 5,000, and 10,000 cells were formed and then imaged. The spheroid diameters were measured using an automated code on MATLAB and showed low variability within groups and statistically significant differences between groups. A recent study by Grosser et al. Has proposed that the formation of a supracellular actin cortex can allow a cluster of cells to migrate as a collective unit. However, we show through F-actin staining that there is no evidence of a difference in the cytoskeletal organization of cells invading in low and high collagen densities. In the figure, you can see that AF568 phalloidin, which is our F-actin stain, is in green. In light of these observations, we hypothesized that the switch in migratory phenotype is driven by physical confinement from the extracellular collagen. To better understand the mechanical and structural properties of collagen, we performed the unconfined compression test on samples of 1 through 4 mg per ml collagen gels. You can see in figure A. The gels underwent 5 steps of compression, each with a magnitude of 3% of the gel's original height. Each compressive step was followed by 3 minutes of hold time. The forces generated during compression increased as collagen concentration increased. We found that the equilibrium stress-stretch behaviors are nearly linear up to 15% compression and highly sensitive to collagen concentration. This allowed us to fit a linear neo-Hookean material model to obtain the bulk shear modulus. The observed trend in bulk stiffness mirrors the trend seen in the collagen fiber density observed under multiphoton imaging, which is the figure all the way on the right. Future work seeks to develop a new mechanical model that combines both structural and mechanical data to explain the physical role of collagen density on breast cancer spheroid invasion. In the future, such a model will allow us to incorporate other features such as collagen alignment and cross-linking between fibers that are known to play key roles in breast cancer progression. We thank the Department of Bioengineering for organizing the undergraduate research program and Dr. Ben Porter for his guidance and support. We would also like to acknowledge the Barner Lab for their assistance with the mechanical testing experiments. Thank you.